In this final section, we're going to discuss the process of fermentation. When there is no oxygen available to act as a final electron acceptor, organisms can still break down glucose to release energy through a process called fermentation. Fermentation begins just as cellular respiration does with glycolysis. As we've seen before, glycolysis results in 2 pyruvate molecules and 2 NADH molecules. Without an electron acceptor, the energy stored in these molecules can't be used. So in the previous section, we talked about how when NADH is used in oxidative phosphorylation, it can be used because molecular oxygen is there to accept the electrons. If the molecular oxygen isn't there as an electron acceptor, the energy stored in these molecules then can't be used. In that case, the net energy gain then is just the 2 ATP molecules. 2 were invested to provide the activation energy and 4 were produced, netting 2. There's no electron acceptor because there's no oxygen. Okay, so once this pyruvate is produced as a result of glycolysis, it still needs to be cleared from the cell. And the NADH, since it can't be used in the oxidative phosphorylation step of respiration, needs to be converted back to NAD+ to begin another cycle of glycolysis. The process of doing this is called fermentation. No additional energy is released during this process. There are many types of fermentation. The 2 types of fermentation we will focus on are lactic acid fermentation and ethanol fermentation. So we'll take our pyruvate through the fermentation process. We will convert our NADH back to NAD+, and either 2 lactic acid molecules or carbon dioxide and 2 ethanol, depending on the type of fermentation. Fermentation breaks down the products of glycolysis so that glycolysis can be repeated with another glucose molecule. 1 glucose molecule yields 2 ATP, 2 pyruvates, and 2 NADH. These are the input to the fermentation stage of anaerobic respiration. The pyruvates and NADHs are fermented, so I can put NADH and pyruvate into my fermentation process, and they're fermented into 2 NAD+ and either lactic acid or carbon dioxide and ethanol. The result of the combined steps of glycolysis and fermentation is the input is 1 glucose, 2 NAD+ and 2 ATP molecules. The output is 2 NAD+ and 4 ATP molecules for a net gain of 2 ATP. In addition, lactic acid fermentation results in lactic acid. Ethanol fermentation results in ethanol and carbon dioxide. The process started with 2 NAD+ molecules, which were recycled by fermentation to finish with 2 NAD+ molecules. Cellular respiration versus fermentation. The big difference is that for each glucose molecule, aerobic cellular respiration yields 30 to 32 ATPs, while fermentation yields only 2. In aerobic respiration, about 55% of the energy in a glucose molecule is used to make ATP. The remaining 45% is lost as heat. In fermentation, only about 3% of the energy in glucose is used to make ATP. The remaining 97% of the energy is lost as heat. In both cases, there is an increase in entropy. No biochemical reaction is 100% efficient. A reaction is more efficient if there is a smaller increase in entropy and more low entropy energy available for cells as a result. Here are some examples of fermentation. Some anaerobic bacteria rely solely on fermentation, such as lactobacillus, which is used to make cheese and yogurts. The alcohol in wine, beer, etc. results from yeast. Yeast is a facultative anaerobe, which just means that it makes ATP by the aerobic respiration we discussed previously if oxygen is present, but is capable of switching to fermentation when oxygen is not present. So the alcohol results from yeast undergoing ethanol fermentation. Bread rises due to the release of carbon dioxide bubbles by fermenting yeast. Your muscles burn after a strenuous workout because they can't get enough oxygen. And so remember, oxygen is needed for cell respiration. So instead, they perform lactic acid fermentation, and that lactic acid buildup in your muscles is what results in soreness. Oxidative phosphorylation in prokaryotes. Prokaryotes may use the electron transport chain via oxidative phosphorylation to yield more ATP. For example, Mycobacterium tuberculosis, the organism responsible for causing tuberculosis TB in humans, is an aerobic bacterium that uses oxygen as its final electron acceptor. There are some differences, though, in how this occurs. In prokaryotes, the electron transport chain takes place in the prokaryotic plasma membrane. In eukaryotes, it occurs in the mitochondrial inner membrane. Aerobic prokaryotes use oxygen as a terminal electron acceptor, while anaerobic prokaryotes use other molecules, sulfur, carbon dioxide, nitrogen, nitrate, etc. Eukaryotes only use oxygen as a terminal electron acceptor. In prokaryotes, the passage of electrons is accompanied by the movement of protons across the plasma membrane, creating the proton concentration gradient. This occurs in the mitochondrial inner membrane of eukaryotes.